Okay, we're back again for the second part of this protein synthesis video. We left off and we said transcription was complete. So this process that we've got here is transcription, where the DNA is copied into mRNA. A couple of little bits that might not have mentioned. We said that the polymerase is responsible for forming the phosphodiester bonds between the nucleotides on mRNA. And we said that the mRNA was complementary to the template strand. Now, actually the mRNA really is a copy of this strand here, the coding strand, or sometimes referred to as the sense strand. Okay, obviously the coding strand is complementary to the template strand is complementary to the mRNA, and therefore the mRNA is a copy of that coding strand. Oopsie daisy. So RNA polymerase catalyzes condensation reactions between the RNA nucleotides, joins them together with those phosphodiester bonds, forming a new polynucleotide chain of mRNA. Then we said that we've got this splicing, the removal of the introns from the pre-mRNA to form mature mRNA. And then we said that mRNA leaves the nucleus via those nuclear pores and moves into the cytoplasm. Now, in the cytoplasm, brings us to translation. So here we've got that second process, translation, where the code on the mRNA is going to be translated into a sequence of amino acids. Now, this takes place at the ribosome. Okay, so the ribosome might be ribosome free in the cytoplasm, or it might be attached to the rough endoplasmic reticulum. Either way, the mRNA strand, this is the mature mRNA strand, binds to the ribosome. Or perhaps it's better to say that the ribosome binds to the mRNA. And the mRNA moves through the ribosome 3 bases at a time. And if you remember, those 3 bases are what we refer to as a codon. Okay, so a codon, 3 bases, and they code for 1 amino acid. So on DNA, there are triplets. On mRNA, there are codons, and they both code for 1 amino acid. So, the ribosome binds to the first, it would be over this end, sorry, the first codon on the mRNA strand. And as it binds to it, there are 2 binding positions in the ribosome, and that allows for 2 tRNA molecules. So these are tRNA molecules. They're not drawn in their cloverleaf shape here, they're just drawn simply, and that allows the 2 tRNA molecules to enter the ribosome. Now those tRNA molecules, they have anticodons. Okay, those anticodons, where we can see this codon here is complementary to this anticodon here. This codon is complementary to this anticodon. So those complementary base pairing again. So complementary base pairing between the codons on mRNA and the anticodons on tRNA. 2 of those tRNAs can enter the ribosome, and they carry with them an amino acid bound to that amino acid binding site. A fairly typical question about tRNA might be, what is the minimum number of tRNA molecules? Okay, and that would be 20, because there are 20 amino acids. Anyway, so that anticodon determines which amino acid can be combined to the tRNA binding site. So if we remember back to the diagram of the tRNA, here we can see there's the anticodon, there's the tRNA binding site, sorry, the amino acid binding site on the tRNA. So as the tRNAs enter this binding site, that allows for a peptide bond to form between the two amino acids attached to the two tRNAs that are in the ribosome at any given time. So this is a condensation reaction. That forms between the amine group and the carboxylic acid group, carboxyl group on the amino acids, forming these peptide bonds. Now, once that peptide bond has been formed, this bond here between the tRNA in the first binding site, this bond breaks. And that releases this tRNA molecule. And you can see this one here has already been released, as have these two. And then the ribosome will move along to the next codon. Okay, as it moves along to the next codon, the tRNA that's currently in binding site number 2 will move into binding site number 1. This one will move into binding site number 2, and a new peptide bond can form by condensation reaction. That would then release this amino acid from the tRNA. That tRNA goes off and picks up another amino acid, and the ribosome moves along one more codon. So the ribosome moves one codon at a time until eventually it reaches a codon which is referred to as a stop codon. And when it reaches that stop codon, the ribosome detaches from the mRNA, and then it may go back to the beginning and start that again. Or what you might find is that many ribosomes attach to the same mRNA strand. So many of these polypeptide chains, now we've got these, this is the formation of a polypeptide chain. Many of those polypeptide chains could form all at the same time. Okie doke. So that process of translation is described here. It mentions that the tRNA with a complementary anticodon binds to the first codon in the ribosome. The ribosome has 2 binding sites, so 2 tRNA molecules combined. Peptide bond forms during a condensation reaction. First tRNA then moves away. That's what's happening with these. And after these tRNAs move away, they go and collect more amino acids from what's referred to here as the amino acid pool. And that process of binding a new amino acid is called activation. And that activation requires, probably guessed it, ATP. Okay, so protein synthesis itself, for a number of reasons, is quite reliant on a supply of ATP. one of those processes that requires ATP is activation. Okay, so mRNA moves along one codon at a time. After that codon's been read, a new tRNA enters, etc., etc., until it reaches the stop codon. And then the specific polypeptide has been produced. At that point, perhaps that polypeptide moves into the Golgi apparatus, it gets folded, maybe it's added to other biological molecules. At the Golgi, it forms its secondary, tertiary, potentially quaternary structures. All of that will be determined by the sequence of amino acids. So obviously the sequence of amino acids determine the bonding, the hydrogen bonds, ionic bonds, disulfide bonds, etc. in the tertiary structure, and the hydrogen bonds in the secondary structure. And that sequence of amino acids is determined by the codons on the mRNA, and we also know that the codons on the mRNA are determined by the triplets present on the DNA. Okay, so ultimately it is the DNA that determines which specific polypeptide is produced. Excuse me. And that's that. What I would say, it's fairly typical questions that you might get relating to this, other than the process itself, and obviously it's really important to learn that process. The other really important skill that you need to learn is completing tables such as this. Okay, so here we have, let's see if I can copy this. Here we have a table. Can I copy that table? No, it won't let me. Here we have a table showing a number of different amino acids. Here is the DNA triplet code, and you're asked to complete the mRNA codon. So obviously the triplet code is complementary to the mRNA codon. mRNA codon is then complementary to the tRNA anticodon. So this glycine, the triplet code is CCT, so cytosine, cytosine, thymine. mRNA anticodon will be guanine, guanine, adenine. So one more, methionine, which is thymine, adenine, cytosine, is complementary to, well, T is complementary to A, adenine, but here we can see in the mRNA that uracil replaces the thymine that would be complementary, so it's adenine, uracil, guanine, and here this would be uracil, adenine, cytosine, so UAC. Fairly typical. The important thing to recognise when you're doing this is to determine what is your starting point and what is your final point. You know that the DNA is complementary to the mRNA, mRNA is complementary to the tRNA. You may be asked, you may be given the codon and you're asked to determine the triplet code. For example here, so here's the mRNA codon, so that is going to be thymine, cytosine, guanine. You may be given the tRNA and asked to determine what the DNA is. Well, they will be exactly the same unless, like in this case, with uracil replacing thymine. And that's that for this video. I hope that's helpful. Just remember to drop me an email if you have any questions. Thanks for your time.